Welcome back to the Deep Dive. And today, we've got a really specific mission. This one's for you, Mr. Martin's Grade 8 Science class.
That's right.
We're aiming to give you, well, kind of the ultimate shortcut to getting the basics of experimental design right.
hmm
We're talking scientific language, those essential variables, and how to build a really strong hypothesis.
Exact. And this isn't just about, memorizing words. We're looking at the structure. It's like learning a secret code, honestly.
A secret code.
Uh-huh.
How so?
Well, once you get this structure down, how to talk about your experiment clearly, what you're changing, why you expect something to happen, your experiments become, well, reliable.
Okay.
People can understand them, they can repeat them, and they can trust the results. And that's true whether you're in your classroom or, in a lab anywhere else in the world.
Okay, that makes sense. Let's unpack this then. Starting with that idea of a universal language. Why the big fuss about shared words and units? Can't I just measure things my own way?
Huh? Well, you could, but the big deal is really about precision. And sometimes avoiding, well, disaster, frankly.
Disaster, yeah.
Oh, yeah.
Yeah.
Think about it. If one team designing, say, a spacecraft uses feet and pounds, and another team uses meters and kilograms, and they don't convert perfectly,
Uh-oh. I sense a story here.
you got it.
Yeah.
The Mars Climate Orbiter back in 1999, a classic example. It was lost, likely because one engineering team used English units, pound-seconds for force, while another assumed metric units, newtons. That little mix-up had huge consequences.
Wow, okay, so clarity isn't just like nice to have, it's absolutely essential for things to actually work.
Precisely. And that's why we have the SI system, the System International. It's the agreed-upon global standard.
Right, the metric system, basically.
Essentially, yes.
Yeah.
It ensures scientists everywhere are measuring length in meters,
Uh.
mass and kilograms, kg.
It's time in seconds.
Exactly.
Yeah.
Energy in joules.
Yeah.
J. It means everyone's literally speaking the same measurement language. No more lost space probes due to unit confusion, hopefully.
And it's kind of cool that some units, like the Newton N for force, or the volt V, or Hertz for frequency, are named after scientists.
It is. It connects you right back to the history of discovery. Now, using SI is one thing, but you also need the right tool, the right apparatus for the job, and the right scale of unit.
Right. You wouldn't measure the thickness of your hair with a meter stick.
Exactly. Or the length of a soccer field with a tiny ruler meant for, I don't know, insects.
You'd use a micrometer for the hair, maybe a regular ruler or tape measure for your desk, and kilometers for measuring the distance between towns.
Perfect. Scale matters. And the type of data you collect matters too. We usually talk about two main types of measurable quantities.
Okay, what are those?
We have continuous data and discrete data.
Continuous. That sounds like something that, well, continues.
Kind of, yeah. Continuous data is something you
Yeah.
measure, and it can take basically any value within a range. Think decimals.
Like height. I could be 1.65 meters tall, or 1.652, or 1.6523.
Exactly. Height, length, mass, time, temperature. These can all be measured with increasing precision depending on your tool. They exist on a continuum.
Got it. So what's discrete?
Discrete data is different. It's stuff you count or group into separate categories. Things that can only be specific values, usually whole numbers.
Like the number of students in the class. You can't have half a student.
Perfect example.
Mm.
Or the number of leaves on a stem, your shoe size usually whole or half sizes, right? Or eye color, which falls into distinct categories like blue, brown, green.
Okay, continuous is measured, discrete is counted or categorized. Makes sense. And you mentioned some key vocabulary earlier, things like variable, apparatus.
Right, and also words like anomaly, which is an odd result, reliability, meaning can you get the same result again, and validity, meaning does your experiment actually test what you think it tests. We'll touch on those.
Good list. But you said variables are really the heart of it all.
Absolutely. Variables are the core of designing a fair test. They determine what you change, what you measure, and crucially, what you keep the same. Get the variables wrong.
And the whole experiment is basically useless.
Often, yes. Or at least the results become really hard to interpret reliably. It's not a fair test anymore.
Okay, this sounds important. Let's break down these variables. You said there are 3 types.
3 essential types for a fair test. Let's think about. Testing which type of cup keeps a hot drink warm the longest.
Okay, I like that example. What's the first type?
The first is the independent variable, or IV. This is the one thing you, the scientist, deliberately choose to change. It's the presumed cause.
So in the hot drink example, if I'm testing a plastic cup versus a ceramic mug versus a metal thermos, The IV
The
is...
type of cup material.
Yeah,
That's the factor you are intentionally varying.
got it. I changed the IV on purpose. What's next?
The dependent variable, or DV.
Yeah.
This is what you measure to see if the change you made had an effect. It depends on the independent variable. It's the effect or the outcome.
Okay, so for the cups, I'd measure how long the drink takes to cool down, or maybe the temperature after, say, 10 minutes.
Exactly. The time taken for the drink to cool to a certain temperature, or the temperature change over a set time. That's your DV. That's the data you record.
Using SI units, like minutes or seconds for time, degrees Celsius for temperature.
Perfect. Now the third type, and this is maybe the most critical for fairness. Controlled variables, the CVs.
Controlled, meaning things I need to keep, well, under control. Keep the same.
Precisely. Controlled variables are everything else that could possibly affect the outcome. The DV that you need to keep constant across all your tests.
Why is that so vital?
Well, imagine testing the cups. If you put boiling water in the ceramic mug, but only warm water in the plastic cup.
Then if the ceramic mug stays warmer longer, I wouldn't know if it was because it's ceramic or just because the water started hotter.
Exactly.
Oh, yeah.
You've introduced another changing factor.
Yeah.
So for the cup experiment, your CVs would be things like...
The starting temperature of the drink.
Yeah.
Gotta be the same for all cups.
Mhm.
The amount of drink, the volume, or I place the cups. Like not putting one in a sunny spot and another in the shade.
All crucial. Keeping CVs constant ensures that any change you see in the DV is actually due to the IV you changed and nothing else. That makes your experiment valid.
Okay, let's try one more quick one. Testing out which paper towel brand is most absolvent.
All right, 4.
The brand of paper towel. That's what I'm changing.
Good. DV.
What I measure.
What? Maybe the amount of water absorbed, like in milliliters.
Excellent. And key CVs.
Okay, got to keep the size of the paper towel piece the same for each brand.
Yes.
The amount of water I start with to test the absorbency, maybe the temperature of the water too.
All good controls. And here's a tip for when you write up your experiments, Mr. Martin's class. Don't just list your controlled variables. Describe how you kept them the same.
So instead of just saying controlled temperature, say kept the water at room temperature, approximately 20 degrees Celsius, for all tests.
Exactly. And briefly explain why to ensure a fair test is usually the reason.
Okay, variables nailed down. We know what to change, what to measure, what to keep the same. What's the third piece? The third cornerstone is crafting a powerful hypothesis.
Right, the prediction. But you said it's not just a random guess.
Absolutely not. A scientific hypothesis is a focused, clear, testable statement, and critically, it has to be reasoned. It's based on some existing scientific understanding or observation.
Okay, so how do we structure it to make sure it's all those things?
The best, clearest structure, especially when you're starting out, uses 3 parts. If, then, because.
Then, because. Okay, break that down.
Sure. If introduces the change you're making to the independent variable.
Okay.
Then predicts what will happen to the dependent variable as a result. It needs to be specific.
Right.
And the because part is your scientific reasoning. Why do you think that relationship between the IV and DV will happen? This is based on what you already know from science.
Let's try it with an example. Maybe something about plants and water? Like, does more water make a plant grow taller?
Great example. How would you structure that hypothesis?
Okay, if
Yeah.
the IV changes. So, if the volume of water a plant receives increases,
Good start.
the DV will change. Then the plant will grow taller.
Specific prediction. Good. Now the crucial part.
Because, the scientific reason, because plants need water for photosynthesis, which makes their food for growth and also to keep their cells firm.
Excellent. See how that because links it back to actual biology, shows you're not just guessing, you have a reason based on science. That's what makes it a strong, reasoned prediction.
Hey, that makes so much sense. The because is the justification. It's the science bit.
It really is. And for students looking ahead, you might even predict the kind of relationship. Like, if temperature increases, then dissolving speed increases because that implies a direct relationship. Or sometimes it might be inverse. If pressure increases, then volume decreases because.
Getting a bit more advanced there. And you mentioned sources earlier. If my because comes from something I read.
Yes, absolutely essential. If you base your reasoning on information from a textbook, a reliable website, or a scientific article, you need to say where you got it from. That's called citing your sources. It gives credit and shows your research.
Okay, so let's put it all together now. Language, variables, hypothesis, the whole cycle. Let's use another classic. Does a plant grow faster with more light?
Okay, great one. Walk us through it. First step.
Define the language and the variables, okay? My 5e, the thing I change, is the amount of light the plant gets. Maybe I measure in lux with a light meter.
Good. Using SI units, potentially, what's the DV?
The dV is what I measure. The plant's growth. So maybe the change in height in centimeters or the change in mass in grams or even counting the number of new leaves.
All good options for measuring the effect. And the critical CVs.
Right. Got to keep things fair. So the CVs would be the type of plant, use the same species, the amount of soil, the size of the pot, the amount of water each plant gets. You got to keep all those the same.
Perfect. You've defined your terms and your variables. Now the hypothesis using our structure.
Okay. If the amount of light is increased, then the plant will grow taller, gain more mass produce, more leaves, DV. Because increased light intensity allows the plant to perform more photosynthesis, producing more glucose, which is the energy needed for growth.
Boom. There it is. The whole setup. You've got clear language, defined variables for a fair test, and a reasoned, testable prediction. That's the foundation of solid scientific investigation.
It really ties it all together. The clear language helps everyone understand, the variables make it fair, and the hypothesis links it to actual science knowledge.
That's the core takeaway. Precision in your language, fairness through controlling variables, and grounding your predictions in knowledge. This whole process, this structure, it empowers you, the student, to move beyond just following recipe-like instructions.
Yeah, it lets you actually design your own investigations, right, and understand experiments you read about.
Exactly. You can analyze them critically.
So for everyone listening, especially Mr. Martin's class, mastering this process really is like getting that secret code. It unlocks how science works.
It really does. And maybe one final thought to chew on. Something that connects to words like reliability and validity.
Okay.
Even if you design a perfect experiment, clear language, fair test, solid hypothesis sometimes, you get an anomaly, a weird result, one data point that just doesn't fit the pattern you expected.
Like one plant under highlight just doesn't grow well, even though the others did.
Exactly. So the provocative question is, if you get an anomaly like that, how does understanding all this stuff, variables, controls, reliability, help you figure out what happened? Was it just a mistake? Maybe you measured wrong, unreliable?
Yeah.
Or did you accidentally change a control invalid? Or could it be that you've stumbled onto something genuinely new and unexpected about how that plant responds to light?
Whoa, that's a deep question. It moves from just doing the experiment to really thinking about the results.
That's the goal, thinking like a scientist.
Well, that's a fantastic place to wrap up. To Mr. Martin's class, thank you so much for diving deep with us today. We really hope this helps you lock down these fundamental concepts.
Good luck with your experiments.
And for everyone listening, make sure you hit that like button and subscribe so you don't miss our next deep dive.